Have you ever wondered what happens when ancient viruses trapped in Arctic ice finally see the light?
Imagine a world where microbes from the last ice age awaken as our climate warms. Are we ready?
It's not science fiction. In 2016, thawed reindeer released anthrax spores and reminded us nature stores surprises.
Join us as we unpack the real risks and myths around so-called zombie viruses and how they connect to climate.
Welcome to Your Pack. I'm Alex, curious about nature's secrets and ready to explore permafrost mysteries with you.
And I'm Blair, here to keep us honest, with evidence and context, as we dive into ancient microbes.
Before we begin, yes, our voices are AI generated, but they reflect our genuine passion for conservation and transparency.
We'll outline what permafrost is, how it preserves life, and why warming releases more than just carbon.
Along the way, we'll bust myths about zombie viruses, discuss real cases, and invite your questions and stories.
Think of this as a journey, a hook, a deep dive, a debate, real-world examples, community spotlight, and hopeful close.
Permafrost is ground that stays below freezing for at least 2 years and covers a quarter of the northern hemisphere. Its icy layers act like nature's cryobank, freezing plants, animals, and microbes for millennia, preserving them as time capsules. Because these regions lack oxygen and stay dark, decomposition slows, letting viruses and bacteria survive tens of thousands of years. French virologist Jean-Michel Claverie even revived giant viruses like pitovirus and mollivirus from layers around 30,000 years old. When permafrost thaws, microbes decompose organic matter, releasing carbon dioxide and methane, which in turn accelerate warming. This creates a feedback loop. More warming leads to more thaw, more greenhouse gas emissions, and further warming. Recently, scientists revived 13 viruses from Siberian permafrost samples up to 48,500 years old. These are giant viruses that infect amoebas, not humans or animals, reminding us to separate headlines from actual risk. Claverie warns that viruses infecting amoebas share features with those infecting animals, so caution is warranted. In 2016, an unusually hot summer thawed a reindeer carcass, releasing anthrax spores that killed reindeer and one person. Influenza and variola DNA has been detected in mummies and corpses, reminding us that a spectrum of pathogens lies frozen. Permafrost covers 25% of the northern hemisphere, but is warming about 4 times faster than the global average. As shipping, mining, and tourism increase in the Arctic, more people disturb frozen soils and risk exposing buried microbes. Microbiologist Birgitta Evengard calls permafrost a Pandora's box of unknown bacteria, some possibly antibiotic resistant, urging preparedness. A 2025 study reawakened microbes from 40,000-year-old samples, showing they can form biofilms after months. Why tell this story now? Because permafrost thaw intersects climate change and emerging diseases, making 2025 ripe for discussion. People are fascinated by history and fear the unknown. Stories about ancient viruses blend both powerfully. They also allow us to connect scary science with calls to reduce carbon emissions and support Arctic communities. Our video aims to inspire awe without sensationalism. The biggest threat may be greenhouse gases, not killer viruses. We'll paint scenes of glaciers calving and pandemics past, not to scare you, but to provide context. Next, we'll move into numbers. How many microbes might thaw each summer, and why human activity matters. Spoiler: scientists estimate that up to 4 sextillion microbes could be released each summer in a warming Arctic. Another factor is zoonotic spillover. 75% of emerging diseases come from animals, and migration north could shift exposure. Humans have no immunological memory of ancient pathogens, our immune systems could be unprepared for even small exposures. Laboratory safety is another issue. Although Clavery's team uses amoebas, other labs must follow strict biosafety protocols. Metagenomics offers a safer approach by sequencing viral DNA without reviving viruses, reducing exposure risk for scientists. The 2016 anthrax outbreak was severe, but contained through vaccination, culling, and soil decontamination within local communities. Clavery suggests using safe host organisms because most ancient viruses require very specific hosts or conditions. Many viruses will be inactivated by oxygen and ultraviolet light once they reach the surface, reducing immediate danger. So, while caution is prudent, the real takeaways are the climate feedbacks, the need for surveillance, and responsible storytelling.
Let's start by quantifying just how much carbon lies locked away. About 1,500 billion tons in permafrost soils. That is nearly twice as much carbon as is currently in Earth's atmosphere, meaning thawing could drastically alter climate. As microbes revive and consume this ancient organic matter, they release CO2 and methane, potent greenhouse gases. This feedback loop is why scientists call thawing permafrost a climate time bomb, because emissions accelerate warming. Next, let's examine those revived viruses in detail. Clavery's team cultured 13 giant viruses from permafrost. One of them, dubbed Pandoravirus yedoma, comes from samples nearly 50,000 years old, a staggering timescale. To minimize risk, researchers use amoebas as hosts because these giant viruses infect single-celled amoebas, not humans. Some media call them zombie viruses, but none of these revived viruses can currently infect people. Clavery cautions, however, that viruses infecting amoebas share features with those infecting animals, which means we cannot assume all are harmless. Beyond viruses, bacteria present an immediate hazard. Researchers reawakened ancient microbes that formed biofilms after months. Birgitta Evengård warns that permafrost contains unknown bacteria, some potentially resistant to our antibiotics, a challenge for medicine. At the same time, genetic material from influenza and smallpox has been detected in ancient corpses. Those pathogens haven't been shown to infect modern hosts upon thawing, but their presence reminds us of the variety. Scientists emphasize that infection barriers make a global pandemic unlikely. Many ancient viruses require conditions that no longer exist. Exposure to oxygen and ultraviolet light typically inactivates viruses, reducing their viability on the surface quickly. Instead of reviving viruses, Clavery recommends metagenomics, sequencing DNA to detect viral signatures safely in laboratories worldwide. Even when an outbreak occurs, like the anthrax case, rapid public health responses can contain it. Vaccination, animal culling, and soil decontamination stopped the anthrax outbreak from spreading across the wider region. However, increased Arctic activity, shipping, mining, tourism, raises contact with thawing soils and potential exposure to microbes for workers. Longer thaw seasons due to Arctic warming 4 times faster than the global average exacerbate this exposure. Researchers estimate each summer could release 4 sextillion microbes from permafrost into soil and water. Zoonotic spillover remains a concern. As species move northward, viruses have new opportunities to jump hosts because 75% of emerging diseases are zoonotic. Permafrost thaw intersects broader wildlife health issues. We can't ignore them. Public imagination leaps to global pandemics, but experts stress that localized outbreaks are more realistic given current evidence. In fact, some of the bigger risks might come from bacterial spores or DNA viruses, like pox viruses. Our next myth to bust is that ancient viruses will easily infect us. Most require hosts no longer present. So if you see sensational headlines promising zombie plagues, remember that responsible science tells a different story. When we quote numbers and risks, we'll always provide sources and ask you to pause and verify. It's good practice to check the sources panel, where we link studies and articles for deeper reading. Some comments might worry that lab research itself is dangerous. Remember, labs operate under strict biosafety protocols. Also, viruses would need to cross complex evolutionary barriers to infect humans. Random mutations alone are unlikely. What about bacterial spores? Anthrax spores survive for decades, and thawing carcasses can reseed them in soil. That's why we emphasize not disturbing permafrost and following local regulations when exploring the Arctic. For safety, your curiosity is important, but we also value your safety. Share questions and comments instead of digging up samples. For those who worry about our AI pipeline, we'll be transparent about how we generate voices and visuals and cite sources. Remember, this channel is about amplifying under-distributed science through engaging storytelling. We appreciate your trust deeply. Halfway through our deep dive, let's recap. Permafrost is thawing, microbes are waking, but a global pandemic is unlikely. Pause and think. Which risk concerns you most? Carbon release, antibiotic-resistant bacteria, or unknown viruses? Use the poll on screen to vote and discuss. Your perspectives guide future episodes and research prompts. Let's also talk about climate models. Current models don't fully account for permafrost feedbacks, which raises concerns. The Intergovernmental Panel on Climate Change warns that these feedbacks could tip the climate over thresholds. A One Health approach connects human, animal, and environmental health. Surveillance in northern communities is vital. That means supporting indigenous health services, investing in pathogen surveillance, and monitoring permafrost thaw to stay ahead. It also means reducing greenhouse gas emissions to slow warming and give ecosystems time to adapt. So the data tell us a nuanced story. Ancient microbes exist, but climate change and carbon emissions are the larger threats.
We've covered the basics and the data, but I keep coming back to one question. Why do these stories grip us so much when the actual risk seems low?
It's because they hit that sweet spot of curiosity and fear. We imagine a virus thawing and leaping into people, even though biology says that's unlikely. Our brains love drama.
And we're wired for narrative. Telling you about a reindeer carcass releasing anthrax is more memorable than a chart about methane, right? That story sticks and makes us care.
True. And there's also a cognitive bias called availability heuristic. Dramatic cases like anthrax outbreaks make threats feel larger than data suggest. Our brains short-circuit nuance. We must remind ourselves.
Yet, if we lean only into fear, we miss the opportunity to talk about real climate feedbacks and things we can actually do, like cutting emissions and supporting Arctic science.
Exactly. And my skeptical side wants us to challenge sensational claims, while still acknowledging there are uncertainties and knowledge gaps to explore. That's where you come in with questions.
So let's pivot. I'll ask you, the pack. Do you think science should revive ancient viruses to study them, or leave them frozen? Drop your take in the comments.
I'll admit I'm torn. Reviving viruses can reveal evolutionary insights, but also carries risks if protocols fail or if hype outpaces evidence. Balance is tricky.
Our community often surprises us with thoughtful perspectives. Someone recently asked whether we could sample viruses without thawing them. Metagenomics might be that answer. Tell us your ideas below.
Indeed, sequencing frozen DNA lets us map what's there without ever cultivating it, which avoids the problem of bringing pathogens back. It's safer and still scientifically rich. Would you fund it?
I love that this conversation is shifting towards solutions. Empowering indigenous communities to monitor permafrost could be another path. Imagine a citizen science network across the Arctic. Would you join?
That would generate real-time data and create jobs. Plus, local observers know their land better than distant researchers do. We could amplify their voices here. Tell us if you're from the north.
Another viewer suggested using solar-powered sensors to detect gas release and microbial activity. What other creative ideas do you have? Comment your inventions below. We might feature them in a future short.
Personally, I'm fascinated by how combining AI...
Remember that our next chapter dives into real-world case studies where these ideas meet reality. It's story time, and you won't want to miss it. Picture the Yamal Peninsula in 2016. A heat wave thaws a reindeer carcass, releasing dormant anthrax spores and killing 2,500 reindeer and causing a human fatality. Authorities vaccinated herds, culled infected animals, and decontaminated soil to stop the anthrax outbreak from spreading. It was a localized but stark warning. Nature remembers. We should too. Our second case isn't a disaster. It's a lab story. Scientists reviving viruses like Pandoravirus yedoma to understand ancient ecosystems. They use amoebas, not animals, as hosts. Watching amoebas swell with giant viruses looks like science fiction, yet it tells us how life evolved in extreme conditions. But remember, these viruses do not infect us. Case 3 takes us to Colorado, where researchers awakened microbes from 40,000-year-old permafrost that formed biofilms after months. It surprised scientists and hinted at resilience. What connects these cases is human impact. Climate change and industrial activity increase thawing, while laboratories act as controlled windows into the past. Our choices shape outcomes. Imagine a speculative scenario. A mining crew uncovers a virus similar to smallpox. Within hours, a worker falls ill and triggers alarms. Public health teams rush in with vaccines and containment. This is not a prediction, but a way to illustrate how early detection, vaccination, and global cooperation can stop an outbreak. Our readiness matters more than ancient pathogens. Another scenario flips the script. Thawing microbes accelerate greenhouse gas release, tipping the climate past a threshold. This could trigger cascading effects on ecosystems and economies worldwide. Visualize glaciers calving with captions. What happens when ice locks away viruses and now melts? This opening hook sets the stage for our case studies with you. Historical flashbacks to the Black Death or 1918 influenza remind viewers that diseases have dramatically shaped societies. They provide context for why we care about pathogens in ice. Comparing the Yamal incident to modern industrial farming helps bridge the gap between remote tundra and viewers' backyards. It shows that our food systems are also vulnerable to pathogens. Interviewing scientists like Claverie can demystify lab work and show why researchers are reviving ancient viruses to anticipate future threats. Hearing their voices adds authenticity and trust. Would you tune in? Speculative what-if scenes allow us to dramatize risk without claiming certainty. We can follow a virus across continents and then show containment. This keeps us grounded and hopeful. Finally, we end each case study with actions: reducing emissions, supporting surveillance, and staying curious while resisting sensationalism. Let's lead by example and inspire others. Our pack can do this. We also share micro case studies from your comments, like a viewer linking to an article about microbes trapped for thousands of years. Your research tips help shape our investigations. When someone shares a pun about viruses being unthawed, we laugh, but also use humor to make complex science accessible. It lowers the barrier and invites newcomers. Our case studies section isn't just facts. It's a conversation with you through polls, quizzes, and calls to upload field notes. We're building a citizen science community. If you've ever spotted unusual footprints on a hike or have local knowledge about changing landscapes, we invite you to share them. Field data enriches our case files. These real-world examples anchor our discussion and prepare us for the final chapters, where community and future come into focus. Stay tuned for shout-outs and innovations ahead. We love shining a light on you, the pack. Recently, at Richard GY1PQ offered to send photos of footprints from Gippsland so we could verify Tasmanian tiger evidence. Your curiosity fuels our research.
We explained that extra toes often come from double-registered steps, dew claws, or soft ground. But we loved the enthusiasm and asked for time codes to investigate more. Keep sharing your finds.
Another viewer joked about dunking ants to determine gender. We laughed, but used the moment to explain how science actually sexes ants through genetics. Your humor keeps the mood light.
During a debate about de-extinction, Fract Alchemist asked whether reactivating dormant genes in chickens could recreate dodos and even dinosaurs. We responded with questions about welfare and ecological impact.
Our reply emphasized that success isn't just the first hatchling, but a self-sustaining population. We asked whether folks preferred gene editing or activation. Tell us in the comments which you support.
We also celebrate milestones together. When our AI pet emotion translator videos released, your excitement and caution made for rich discussions. Balancing hope with ethics is our collective job.
Some of us asked about the ethics of AI hosts. We openly share our process and keep improving, so our voices feel real and trustworthy. Your feedback guides our transparency.
When @Musketeer Chris sent a pun about pigs doing karate, pork chop, we let out a howl and promised to include more viewer puns of the week. Keep them coming. We adore the creativity.
We shout out @Kindred Creatures for thoughtful pushback on de-extinction and for asking us to clarify ethical guardrails. Constructive criticism helps us refine our content. Thank you always.
To boost retention, we use viewer names like The Pack and call back to earlier puns or facts. These little Easter eggs build community. Let us know if you catch them all.
We also launched polls asking whether you'd sample a permafrost core. Results were split, showing curiosity matched by caution. We love that balance. Keep voting and sharing why.
As we pivot to the future, thank you for your questions, your humor, your skepticism, and your hope. You make this channel thrive. Keep the conversation going below. Looking ahead, scientists advocate a One Health approach that links human, animal, and environmental well-being to monitor thawing permafrost and emerging diseases. This holistic view helps us prepare. Metagenomic sequencing and remote sensors could become routine, scanning permafrost for viral DNA without reviving pathogens and alerting us to changes. Imagine smartphone alerts for microbes arriving soon. AI can help by analyzing vast climate and pathogen datasets, flagging patterns that human researchers might miss, and recommending targeted interventions. But transparency and ethics are critical for trust. Citizen science will be part of the solution. Imagine school children in Alaska collecting soil samples for sequencing, and indigenous elders guiding monitoring. Empowerment builds resilience. Another hopeful trend is the integration of climate modeling with public health. Forecasting permafrost emissions can inform vaccine stockpiles and emergency preparedness. This synergy saves lives and money. We must also invest in Arctic infrastructure, health clinics, internet connectivity, clean energy, to support communities on the front lines of climate change. Equity matters in adaptation. Share your ideas. Perhaps the most critical innovation is social, reducing greenhouse gas emissions through policy, technology, and individual choices to slow thawing. Vote, innovate, and advocate to make a difference. As we consider tomorrow, remember that fascination with ancient viruses can drive support for climate action. Our curiosity becomes a catalyst for hope. Let's channel it into action together. We covered what permafrost is, why it preserves life, and how warming can unlock both microbes and greenhouse gases. We learned that revived viruses may